In human biology, ceramide acts as the structural and metabolic hub of the sphingolipid economy. It sits at a biochemical crossroads, sensing cellular stress and directing the flow of lipid signaling across the membrane. In a patient with Alzheimer's disease, ceramide heavily accumulates in vulnerable brain regions. As senile plaques form and neural membranes degrade, ceramide stabilizes the enzymatic machinery that generates amyloid, executing post-mitotic neurons through severe oxidative and mitochondrial injury. Because ceramide accelerates this neuronal death, the therapeutic imperative in neurology is straightforward: lower the lipid, either systemically or locally. In an oncology ward, the clinical picture inverts. Here, ceramide operates as a potent tumor suppressor. When targeted radiation or chemotherapy strikes a dividing squamous cell carcinoma, ceramide is the specific death signal those therapies recruit to kill the malignant mass. A successful tumor survives by evolving mechanisms to escape this lipid. Therefore, the therapeutic imperative in oncology is the exact opposite: raise ceramide to restore the death signal to the cancer cell. One single molecule, relying on the exact same underlying chemistry, carries two opposing verdicts. Its presence represents a pathological catastrophe in the aging brain, yet offers the primary mechanism of cellular destruction in a tumor. This divergence is a direct consequence of the physical membrane in which ceramide operates and the mitotic status of the tissue it inhabits. This flowchart illustrates the sphingolipid rheostat, a metabolic gradient setting a cell's fate, with ceramide at the center. When enzymes push the balance left, accumulating ceramide and sphingosine, the cell leans heavily toward programmed cell death. When sphingosine kinase 1 and 2 phosphorylate sphingosine, sphingosine 1-phosphate, the cell biases towards the opposite extreme: survival and aggressive proliferation. This diagram shows the 6 mammalian ceramide syntheses. Ceramide is actually a family of molecules categorized by carbon chain length. These lengths dictate wildly different responses. The 18-carbon chain, from ceramide synthesis 1, acts as a strict tumor suppressor. The 16-carbon chain, from synthesis 6, triggers a protective anti-apoptotic response. Because a 16-carbon ceramide protects a cell, while an 18-carbon ceramide kills it, measuring total ceramide in a bulk lipidomic study aggregates opposing variables. It obscures the highly localized chain-specific signaling that actually determines pathology. This dynamic cross-section models the plasma membrane's resting state. Floating within the fluid phospholipid sea are lipid rafts, rigid nanoscale assemblies packed with cholesterol and sphingomyelin. Under stress, sphingomyelinases hydrolyze the sphingomyelin, cleaving its head group to leave behind ceramide. This new ceramide, intensely hydrophobic, forcibly displaces the surrounding cholesterol. These ceramide-dense regions rapidly snap together, coalescing into massive, rigid structures known as ceramide-enriched platforms. This massive structural reorganization acts as a general-purpose signaling amplifier. It drags scattered receptors and signaling molecules into extreme physical proximity. By reorganizing the lipid geography, the membrane uses ceramide's physical chemistry to convert a diffuse biochemical whisper into a localized, inescapable command. This split-screen diagram compares how that identical ceramide-enriched platform dictates outcomes in a post-mitotic neuron on the left and a malignant carcinoma on the right. In the neuron, the platform traps the amyloid precursor protein alongside the base 1 enzyme. This cleavage releases amyloid beta, triggering oxidative stress and more sphingomyelinase, feeding forward into a destructive loop. On the right, in the tumor cell, the exact same platform clustering CD95 death receptors commits the cell to apoptosis. To survive, the tumor uses glucoceramide synthase to attach a glucose molecule to ceramide, dismantling the platform and conferring multi-drug resistance. It also relies on overexpressed ceramidescence and SK1 to drain whatever ceramide remains, converting the death signal into the pro-survival mediator sphingosine 1-phosphate. The structural pathology of both diseases hinges entirely on this shared physical stage. Neurology fights to dismantle the platform to halt amyloidogenesis, while oncology fights to rebuild it to restore apoptosis. If the biochemical mechanics operating on the membrane are identical, why does the exact same execution order mean two different things? Ceramide's job is to sense stress at the membrane. amplify it into a platform, and execute the cell. That computation remains static across biology, regardless of the cell type. The divergence lies in the tissue's capacity for replacement. A neuron is post-mitotic. When ceramide executes a pyramidal cell, the surrounding circuit permanently loses a component it can never rebuild. The death signal is a tragedy. A malignant cell is defined by dangerous, unchecked proliferation. When ceramide executes this cell, it applies a necessary biological brake that the host organism has lost. The death signal is a rescue mechanism. Ceramide acts uniformly as an executioner. Its clinical valence, disease versus cure, is dictated entirely by the axis of replaceability, whether the host tissue can afford the death the lipid commands. Because both fields target the same pathway for opposite outcomes, their pharmacological strategies run in perfect reverse. To protect the neuron, neurology relies on neutral sphingomyelinase 2 inhibitors. The objective is to push the rheostat toward survival and halt the exosomal propagation of tau. To kill the tumor, oncology utilizes glucosyl ceramide synthase and SK1 inhibitors. The objective is to dismantle the tumor's escape machinery, weld the rheostat to the ceramide pole, and force apoptosis. This schematic maps the systemic risk. A ceramide-lowering drug protecting the brain reaches peripheral tissues, stripping away a tumor suppressor signal, a pro-oncogenic liability. Conversely, a ceramide-raising oncology drug poses severe neurotoxic liability if it crosses the blood-brain barrier, executing irreplaceable neurons. Because these two systemic pharmacologies are pointed directly at each other, therapeutic success now demands absolute tissue selectivity. Moving this pathway to cure one disease provides the precise biological catalyst to accelerate the other.